Whenever you're ready.
Great. Good evening, everyone. Thank you for joining the Town Hall and the Trusted Messenger campaign. There's a lot of news going on in the last few days with COVID-19. And so let's get started. Dr. Lin and myself are going to talk about COVID-19 and what's going on. So first slide. So these slides were courtesy of Dr. Katie Mullane at the University of Chicago, and we just wanted to give a general overview of what the coronaviruses were. So all of us acquire coronaviruses as children, and children are fundamentally different than adults with their immune system, and they tolerate coronaviruses a lot better. Most of the older coronaviruses have several types of coronaviruses. The older coronaviruses have a stable genome. The genome has a correcting polymerase, so it doesn't make too many errors. Go to the next slide because we'll get out of the MERS stuff. So SARS-CoV-2 is a little different. The four types of coronaviruses are alpha, beta, gamma, and delta, and they affect many different species. You will see alpacas infected, you'll see whales are affected by them, but SARS-CoV-2 is a sister of CoV-1, which caused the original SARS, and MERS, which predominantly is amongst camels. And SARS-CoV-2 starts out with bats, basically. It's a sarbecovirus, and it comes from the horseshoe bat, and the intermediate species was the, what's a little animal? I forgot the name of that animal. That was the amplifying species that was traded in China, and it jumped into the human hosts. It's been circulating probably between 1948 and 1999, and so you need to have an outbreak. You need to have enough of the virus getting into hosts so it can go from person to person, and that's how it jumped into humans and adapted nicely. SARS-CoV-1 died out, if you remember. The pangolin. There you go. I forget about the pangolin. So the SARS-CoV-1 died out because basically it was too virulent of a virus. It didn't have a significant amount of preclinical or asymptomatic phase. So you didn't have a large number of asymptomatic transmission. And I think 800 and some odd people passed away. And this all started out with one party, one basically wedding, and spread throughout the world. Next slide. So so SARS-CoV-2 starts out in the bats. There's a pangolin, and then you had SARS-CoV-1, which died out, and then the pangolin down here with SARS-CoV-2 is spread, and this is going to become an endemic virus that's going to be with us for a long time. If you look at any of the respiratory viral panels that you do in the hospital, you also have HCOV3 and other coronaviruses out there, which
Dr. Chundy, you accidentally muted yourself.
Okay, sorry, we're having all sorts of technical glitches today. So SARS-CoV-2 jumped from pangolin to humans, and in these wet markets has now become endemic throughout the world, pandemic, and it's going to become endemic over the next few years. Can you go to the next slide? There you go. So what's happening now? As we have so much transmission going on, we're developing mutations, and these are called variants. These variants are occurring interestingly, not all over people who get infected. This variant occurs differently because SARS-CoV-2 has the ability to correct itself. And most of the variants occur in people who are immunosuppressed, but not immunosuppressed enough where they pass away quickly. So the virus continues to mutate, and it picks up mutations that allows it to have escape mutations, escape variants. And so if you look at the South African strain, or if you look at the B117 strain from England, the mutations actually occurred in one host or two hosts, and then spread because they had a survival advantage over the main strain that's out there currently. So the SARS-CoV-2 actually mutates fairly slowly compared to influenza, and much slowly compared to like HIV. HIV in a single person will have thousands of mutations in a single human being over their lifetime. So the mutations now, what do they mean? The current strain that's taking over the United States, the B117, which is projected to become the dominant strain by April, uh, is more infectious, and it seems to be more pathogenic than the prior DG614 strain. Next slide. So the more the virus that's circulating, more mutations. The United States accounts for a quarter of the world's infections. So we actually have a lot more mutations in the United States than we know. And the reason why we don't know is we simply have not been looking. The CDC has recently put out, I think, $400 million to get sequencing done on a national level. Illinois ranks very low in the sequencing world. And the original way SARS was figured out in Seattle was because the Gates Foundation actually had a influenza sequencing grant out there, and they figured out that there was a new novel virus that was circulating. It happened to be what we know as COVID-19. So in Chicago, I believe Rush University has a grant for about $5 million for sequencing, and it has to go through a process to vet who needs to be sequenced. And where the goal is to have 5% of all the SARS-CoV-2 sequence, so we have an understanding of how the mutations are going to affect the vaccines and how it's going to affect transmission. Next slide. So the first variant was the East China variant, the DG614 strain. Quickly took over the variant that originated in Wuhan. It basically replaced most of the variant within a few months, and it accounts for about 99% of the US infections. And it has no change and the vaccine is designed for this variant because that was the predominant variant that was circulating at that time. Next slide. So what we call the UK variant is basically a B117 variant, and you will see these variants described by these numbers. The 501YV1, now there's V1, V2, V3 variants, are a significant concern. And the reason is this variant attaches to the ACE2 receptor tighter than the prior G614G variant. It turns about 20 degrees, gets in deeper, and hangs on tighter. And so it makes it it's much more efficiently transmitted than the prior variant. It also has several deletions, a H69 to V70 deletion, and then the Y144/145 deletions. And it helps evade the antibodies that people make. Next slide. So this is the B117 variant, and you can see that it was about 5 to 10% of the strain in the UK, probably around October or so. But now it's become the dominant strain in the UK and accounted for the massive amount of increase that happened in the UK. It grows much better in cell culture. It replicates faster. It's easier to spread, and the death rates are actually higher than the old G614 strain. The current vaccines do actually cover and neutralize the B117 strain, but the big concern is this variant, which is the B117 strain picked up, which is the E484K. And this was a variant of concern, and now it has been shown that it does not, the vaccine is not as effective. We're going to talk about that a little later in our presentation. Onwards, next slide. And so this is the variant, what we call the South African variant, which is the 353 strain, and it has that 501YV2 variant. There, it has 8 to 10 mutations in the spike protein, and it basically binds tighter to the ACE receptor. It also has this E484K, and it does not contain the deletions that the British variant has, which is at 6970 and the 144/145 variant. This was found in South Africa at Nelson Mandela Bay. And they think the reason why in South Africa this has become such a problem is there's a significant amount of uncontrolled HIV population in South Africa, and this allowed for the escape mutation to occur. And they've actually localized it to a small area where this started. But now it's become the predominant strain in South Africa. It has rendered their main vaccine which was the AstraZeneca vaccine, useless. About 10% of AstraZeneca, it only has about, the AstraZeneca vaccine only has about a 10% efficacy. So they started vaccinating everyone with the Janssen or the Johnson & Johnson vaccine. And they've actually dispensed about 30,000 doses of that vaccine because of the outbreak over there. Next slide. This is the Brazilian variant. The Brazilian variant, there was a place, Manaus in Brazil, where the natural experiment was allowed to continue with the infection. And this area is isolated. It only has 2 roads that come in, and everything else comes in by river. And the government there decided that herd immunity is going to be the cure for coronavirus. And what it did was it basically decimated the population over there. So this variant has several mutations. This variant has overtaken the original tragedy that took place over there, because over 70% of the population was infected with the DG614 strain, and they thought, hey, we've achieved herd immunity, we are going to be safe. And this variant came in there, and they have now reinfection rates, and there's a surge since mid-December, and the P1 mutation now accounts for about more than half of their cases. There's a P2 mutation that was isolated from Rio de Janeiro, and all of these mutations now are isolated in the United States. So next slide. This is Danish variant, but it's which was thought to be due to the outbreak in California. But now we have a lot better data that shows there's actually a California variant, a C10 variant, which seems to be the major cause of the outbreak in Southern California. And today in New York, the New York Times, it's unpublished, and this is out of using a software platform from Caltech that identified a new variant in New York, which is responsible for a significant portion of the transmission going on in New York. So as we're sequencing, what are we finding? We're finding that there are more and more and more variants occurring in the United States, and we really can't blame it on Brazil or South Africa or the UK. We're going to end up having lots of variants in this country. And so now we are in a push against time to get as many people vaccinated as possible. If you listen to Dr. Osterholm on television, he is the PhD from who ran the Minnesota Department of Health, and is actually a model for public health, for departments of health. And he's advocating getting as many people vaccinated as quickly as possible because of these variants, with even 1-dose vaccine and then delaying the 2nd dose. That is not the official position of CDC nor most infectious disease doctors, including me. Next slide. So this is a slide from the New England Journal that basically says, how do you test for vaccine efficacy? So basically what you do is you take neutralizing antibodies and you do a monkey kidney cell line, and you see is there viral replication or not with the serum that contains the vaccine related antibodies. If there's no viral replication, you know you have an effective vaccine. If there's viral replication, you know the vaccine doesn't work against that strain. So this is being tested on a regular basis from vaccinated and unvaccinated people in a cellular model, and that's how we test for vaccine. Next slide. So Dr. Lin's going to talk about pregnancy lactation, because this is a major issue, and both ACOG and Society for Maternal Fetal Medicine have really good guidance on this, and it's really important for most of the docs to be able to advise their patients. Dr. Lin.
Thank you, Dr. Chundy, and I thank the Medical Society for putting on this event for this evening. May I have the next slide, please? So I think we have to just step back and realize that strategy of vaccination has been one of the greatest public health achievements in the 20th century, and actually it began in the 18th century. We certainly have lived in an era where many communicable diseases which cause routine death have been controlled through immunization programs. And I think that we also have to realize that the COVID infection in a pregnant woman is not something which we can ignore. There's increasing number of case reports and small series showing that women who acquire the infection while pregnant have a worse course, more admissions to the intensive care unit, more severe illness, as well as there appears to be an increase in preterm delivery from the to babies. Data also has been accumulating that we can now demonstrate both virus in placental tissue and transplacental transfer of both virus and antibodies. And I'll touch on the latter at the end of my comments. One thing we have to stress to our patients that the two vaccines that we currently have on the market, Moderna and the Pfizer products, do not cause COVID infection. We are also faced with the reality that during the clinical trials, pregnant women were not routinely admitted into these trials. And so we are dealing with our recommendations without adequate type 1 data from controlled trials, both in terms of efficacy as well as safety. But the American College of OBGYN and the Society of Maternal Fetal Medicine, and most recently, the World Health Organizations have come to agreement with the following statements. May I have the next slide, please? So it is recommended that COVID-19 vaccines should not be withheld from pregnant individuals who meet the criteria. And I'm speaking about the ACIP recommended priority groups. And so if patients desire the vaccine and are offered it, we are recommending that this be the correct approach right now. Clearly, as I mentioned before, we don't have a lot of safety data. It is now being accumulated. There are beginnings of clinical trials which are actually including pregnant women, as well as lactating women in these studies. But women should be free to make their own decisions in conjunction with their clinical care team. One thing I want to stress, and this has been stressed by the American College, this should not be a prerequisite. So if a woman who is in the appropriate risk group is offered the vaccine, she shouldn't delay receiving the vaccine because she hasn't had time to sit down with her clinical care team. May I have the next slide, please? When deciding whether this is an appropriate strategy to take or not, we recommend that we look at other variables, such as the level of activity of the virus in the community. Again, emphasizing the potential efficacy of the vaccine. There is nothing biologically that suggests that the vaccine would not be effective in a pregnant woman. And we do know that the infection itself has been shown to have risk and severity of both maternal disease and probably effects on the newborn. And there is also nothing biological to suggest that these vaccines won't be safe. Again, we don't anticipate this being a mutagenic. It's not an active virus. It's not a live virus. We certainly vaccinate many women for other conditions while they're pregnant, including influenza and the bacterial vaccinations for Tdap, because the benefits seem to outweigh the potential risks. May I have the next slide, please? Similarly, there appears to be no justification to withholding the vaccine in lactating women. We do know that the virus, if a woman is infected, can be detected in breast milk. There hasn't been any associated clinical significance to this at the moment, but that ACOG recommends that the vaccine be offered again to lactating individuals as similar to non-lactating individuals when we meet the proper criteria. And we're faced with the same conclusion that the theoretical concerns regarding the safety of the vaccine do not outweigh the potential benefits. So in summary, we're trying to avoid forcing pregnant lactating women to be at risk for the infection, because we think that the infection poses a greater threat to their well-being than any vaccine that we've been talking about. Next slide, please. Another big issue has to do with fertility. This has been looked at, and to the moment, there doesn't appear to be any reason why fertile women who are contemplating pregnancy avoid getting vaccinated. And again, we should encourage women who are not pregnant, even though they may be anticipating trying to become pregnant, there's no reason to avoid the vaccine. Similarly, if a woman does conceive, and let's say she received the first dose, the second dose should be indicated during the normal time sequence, and there shouldn't be an attempt to avoid administering the second dose. In an effort to try to contribute to the scientific knowledge about these infections, we're recommending that our patients consider enrolling in the CDC VSafe program, where they can notify the CDC if they're having any changes related to the vaccine, or if they're having any side effects from it. Next slide, please. So this is fairly recent data from last month. It's not a tremendously large study, but it's an interesting one. And these investigators found that the SARS-CoV-2 IgG antibodies were transferred across the placenta in 72 out of 83 pregnant women who were seropositive for the infection. So these are women who became infected during their pregnancy. As would be expected, IgM antibodies were not detected in the cord blood sera. And the transfer ratios were associated with the time that elapsed from maternal infection and was not related to the severity of infection. So again, from a biological perspective, there's no reason to suspect that this would not be the case from vaccination. And so we actually find the transplacental transfer of the antibodies to be something that we're interested in promoting, because we think that this passive immunity may provide neonatal protection from this infection. And again, it's a small study, it's the beginning. There are other studies now that I know have had similar observations, but I think it's encouraging. And it's encouraging to know, I think, that we're on the right track, and that as we accumulate more data, we'll be a little bit more convinced. But at the moment, again, the infection appears to be a greater threat, and a real threat, than any theoretical concerns that we may hold regarding the use of these RNA vaccines in this reproductive age population. Again, thank you for your attention, and I'll turn things back over to my colleague, Dr. Chundy.
So we're going to talk the next part about the Johnson & Johnson vaccine and why all of this. You're going to hear announcements tomorrow about how the federal government is going to change the vaccination process. And this is in response to what is considered a race against time currently. Next slide. So our objectives are to talk about the epidemiology of the pandemic and the current state of vaccine rollout and immunity, and then the real world data on vaccine efficacy, which was published in the New England Journal, and how the variants are playing the role in the epidemic. And then we're going to talk about the Janssen vaccine, now owned by Johnson & Johnson vaccine. Next slide. So this was published, actually, FICOR is a group that came up with the original modeling, September, October, of vaccine efficacy and then herd immunity, and where we will reach herd immunity. And these were based on several assumptions. So based on these assumptions of vaccine efficacy of 50%, 60%, 70% or higher, it would figure out where you will reach herd immunity, what level of herd immunity we were required to decrease the amount of transmission to less than 1 and put an end to the pandemic, which is what we are trying to do. So this is the current FICOR modeling. This was actually an article from the New York Times, which actually has the best graphs out there for this. So this is an estimate to path to herd immunity. The blue range that you see up there is where the herd immunity is. The orange color here that you're looking at is people who've been infected, which we think is somewhere between 20 and 30% of the population. Close to 100 million people in the United States, 80 to 100 million people, if you take the low end or the high end. And this is the fully vaccinated now, which are about 8 million. You've had about 60 million doses of vaccine now with 1 dose of vaccine. And so if you do this, by July, this is an estimate of people who get herd immunity from natural infection. And if you take the more fully vaccinated, that means how many people, where we will get to for fully vaccinated at the current rate of 1.7 million doses. We'll get there somewhere in October, November before next winter. Next slide. So what happens if we do 3 million vaccines a day, which is what we need to do? On the prior slide, if we just went down at 1.7 million, the estimated death rate would be well over 120, 130,000. So you're talking about now we have 500,000 deaths, and you know we have 100 plus people on the call today, and if you can ask yourself, would you believe we would lose 500,000 Americans? And almost every one of us knows somebody who's been affected by COVID now. I mean, that's an unfathomable number to me, and I think most everybody else in the physician world. But here we are. So if we do 3 million vaccines a day, the threshold for immunity will be reached somewhere in July, and you'll have about 90,000 deaths by the end of the year. Next slide. So what happens if you have relaxation of mitigation efforts? So if you relax the mitigation efforts, that means you're gonna have most of the bars open, stadiums open, as is happening in New York currently. You know, they're thinking of opening in New York. Then you're going to reach herd immunity much faster by natural infection, the upper line here, by June. That'll cost us about 170,000 people to die by the end of the year. So we have 500,000 now, that'll be 670,000. And vaccine immunity, it means that we're vaccinating at the current rate, will be reached somewhere in October with a significant mortality rate. Next slide. So if we can reach this unlikely figure, if we have enough vaccine, 5 million doses, then the total immunity really increases quickly because vaccine immunity will be reached by May. Okay, and there you'll have a significant drop off of mortality, and that's where Osterholm and company are saying, you know what? 1 dose of vaccine has significant efficacy in all, whether it's the Moderna vaccine, the Pfizer vaccine, and we're going to talk about the J&J vaccine. And you will significantly decrease death. And if that's our goal, that's what we should be doing. The downside of this in the infectious disease world is we may actually be increasing the risk of getting those escape variants that we talked about, the variants of concern, and we may end up having vaccines that will be ineffective because we'll have more resistant viral strains to vaccines. Next slide. So if we remove all precautions, okay, you'll reach the threshold quicker and you'll have a significant amount of mortality by the end of the year, 320,000. If we just open up everything like Brazil did in Manus, this is the mortality that you'll have by the end of the year. And you'll achieve vaccine threshold again by October because you're not increasing the vaccine rate, but you'll achieve the precautions by May. Next slide. By herd immunity by May. Sorry about Now, if you have more infectious virus, like the vaccine variants now, the 117 strain, the South African variant, you will have significant increase in mortality, even with the current way we're vaccinating. And this is why the federal government is going to make announcements in the next day of changing their approach and trying to take control of mass vaccination centers and vaccine distribution and getting vaccine to people's arms as soon as possible, so nobody's holding vaccine. Because of this increase in mortality, you may, by the end of the year, have 200,000 more dead, which would be a loss that's unfathomable. Next slide. So if you have variants, now this is with all the, I'm sorry, go to the previous slide now. So this is with the variants and using all our precautions. That means, still wearing your masks and not having a lot of things open. So you'll have 200,000 deaths with the South African strain or similar variants that will predominate. Next slide. So if you take away precautions, okay, and you have the variants becoming the most common thing circulating, you'll lose another 500,000 people. So this is the extreme scenario of letting everything fly and having herd immunity by April, by natural herd immunity, and you can see the majority of the population will be infected by the end of the year, but the mortality would be over 500,000 people. Next slide. So this is looking at the current behavior and having a fast vaccine rollout. So by the end of April, you'll have the current strain that we have there, the 614 strain going away, and the 117 variant taking over. Next slide. And if you have current protective behavior but no vaccination, the variant will become even a greater risk by the end of April. Next slide. This is from the University of San Diego, UCSD. And if you have a variant out there, and if you relax the protective behaviors, that means the restaurants and everything else that we all want to go back to, the variant becomes a predominant strain by the end of April. So, we're going to be stuck with masks. We're going to be stuck with having socially distanced gatherings for some time. I know everyone's feeling much better now that the rates have plummeted, the amount of people passing away has plummeted within the hospital, but it's not over yet. Next slide. So we now have a new vaccine, and this vaccine is a significant game changer. So why is this vaccine such a game changer? Well, for all of us who have private practices or have clinics with just a refrigerator, well, this vaccine has a long shelf life, over a year. You can put it in your refrigerator, it sits there. It's a single dose vaccine that's currently that's going to be approved in the next day or so. It has a significant better side effect profile that we're going to see. And there are current trials going on giving 2 doses of this vaccine to show that efficacy may be more. Next slide. So what is it? Well, the first time we talked about these vaccines, we said this, the RNA vaccines do not get into your nucleus. Well, this does get into your nucleus. So this is a Adenovirus 26 vaccine. And this platform has been around for some time. This was originally used for, it's a chimpanzee. This is a human adenovirus that was discovered in a 9-month-old child in 1956 from an anal specimen. And then what they did was they basically took away one of the E1 genes by deleting the E1 gene and made it replication incompetent. And this has been the vaccine platform that's been used for Ebola. And so it's been given to over 200,000 people already, this platform. So it is not something that's new. So an AD26 has actually a very low prevalence in humans, and so it has very efficient transduction of the epithelial cells. So it's able to get to the target cells. Next slide. So here's the safety data as of September 4th, 2020, and there's actually a monitoring for this with ADVAC safety database. So over 114,000 individuals have been given AD26 vaccines, mostly with Ebola, and it's been used in pregnancy. It's been used in multiple populations, and most of the reactions are grade 1 to 2 and occur within 24 hours, and almost all of them resolve within 72 hours. This model has been used for HIV vaccine, malaria vaccine, Ebola vaccine, and now it's been used for SARS-CoV-2. So it is a very well-developed platform. Next slide. So what's the mechanism of action? Dr. Petrak, my partner, gave a presentation on this, so I borrowed his slides. So basically, this is the adenovirus. It's a whole adenovirus with the E1 with the gene deletion there, the E1 gene deletion, and you basically take the gene of the spike protein and you put it into the adenovirus. It delivers it into the cell. It gets into the nucleus where it then replicates, puts it into the RNA and then into your mRNA, and the mRNA starts making all of these nice little spike glycoproteins that go sit on top of cells. And then your body, your CD4 cells recognize these spike glycoproteins sitting up there, and they start making antibodies to these with a very good response, an excellent response. So this does use the host DNA to make the messenger RNA to make the spike glycoprotein, which will then present itself to our antigen presenting cells. Next slide. So the J&J vaccine variants, if you looked at these countries where this vaccine was used in the United States, most of the vaccine, you had the G614 variant at that time. In South Africa, there was a significant amount of the South African variant. And in Brazil, there was a significant amount of P1 variant. There was not a significant amount of B117 variant. Next slide. So this was this was submitted to the FDA. You can go on the FDA website of the Janssen AD26 vaccine, and this was the ongoing phase 2 randomized double-blind placebo-controlled trial of a single dose of this vaccine to about 40,000 individuals, and they used vaccine efficacy against a centrally lab confirmed moderate to severe critical COVID-19 in all geographic areas, and the trial showed that about 66.9% were efficacy at 14 days after a single dose, and 66% efficacy after 28 days. And basically, there were no deaths in the vaccine group. And we're going to go through some of these slides. So there were 116 cases of COVID in the vaccinated group versus the placebo group, and then at 28 days, 66 versus 193 cases. Next slide. So what were the populations that were vaccinated were given? 43,783 patients, 22,000 were vaccinated. Look at the age breakdown, very good age breakdown, 18 to 59, 67%. Over 60, about 33%. 65 and greater, 20%, and more than 75, 3%. And then if you looked at the Moderna, Pfizer, and J&J vaccine, you can see that African Americans comprised 9.8% of the 9.8% of the Moderna, 9% of the Pfizer, almost 20% of the J&J vaccine. Hispanics, 20% for Moderna, 24% for Pfizer, 45% for J&J. Obese individuals, again, a nice representation for both Pfizer and J&J, and HIV patients at about 601 of the 22,000 vaccinated people were 43,000 of the people that participated. Next slide. And where was it conducted? It was conducted worldwide. The USA accounted about 44% of the population, the Brazil 16%, so you had variants down there, South Africa 15%, and Latin America 23%. So you had a nice worldwide distribution for this vaccine, for these cases. Next slide. So what were the adverse effects? And this was the really heartening piece of this whole vaccine. If you looked at Pfizer, 80% of the people had pain at the site, and most of us who are physicians here have taken this vaccine, it hurts. So Pfizer and Moderna both had a significant amount of pain at the site, but J&J, 49%. Fatigue, Pfizer, Moderna, 50% and 69%. 38% for J&J. So it has less side effects than both the Pfizer and Moderna vaccine. Headaches were present in half the Pfizer population, more than half in Moderna, 39% in J&J. Fever was present in 16% of Pfizer, 15% of Moderna, 9% of J&J. Lymphadenopathy was found in a significant amount of people in both the Pfizer and Moderna vaccine. Were unable to find any lymphadenopathy with the J&J vaccine. So the other unexpected thing was with the J&J vaccine, Bell's palsy, three in the vaccine group, 2 in the placebo group, same as the expected background. Tinnitus was interesting. Tinnitus was found in 6 in the vaccine group, 1 in the placebo group. The FDA believes these are all not related, but they're looking into that. Seizures were found in 4 in the vaccine group, 1 in the placebo, again, not related. And then each of pericarditis, brachial radiculitis, and that's kind of very strange. It occurred almost immediately at the time of vaccine. And that in that singular case, either you had to inject it into the brachial plexus, which is kind of far away from your deltoid, but it was reported. There were 19 deaths total in this study, 3 in the vaccine group, 19 in the placebo group. None of them were related. There were suicides, there were heart attacks, there were pulmonary emboli. So there were other causes of death, but not related to actual vaccine or anaphylaxis. There was 1 anaphylaxis type reaction reported, and that was it. Next slide. So a very, very safe vaccine. So the efficacy. Now you have to define moderate to severe and severe. These are defined a little differently than what we as infectious disease doctor thinks as severe. So moderate to severe, our heart rate was not very fast. There's not very sick people. The efficacy in the US was 72%, South Africa 64%, and Latin America 61%. In severe cases, that means a heart rate of over 100. The efficacy was still pretty good, 78%, and at 28 days, it was 86%. And the reason for this increase in efficacy is the antibodies tend to pick up as time goes on. In South Africa, 73%, a little decrease in efficacy, and at 28 days, 82%, and in Latin America, 89%. Next slide. So this is important because at day 7, you start seeing the separation from placebo. It really takes till about day 14 when you start seeing the cumulative incidence of critical cases go up in the placebo group. And you can see by day 28, by day 35, you really have good protection. And in the vaccine efficacy here, the heartening thing is you can go all the way out to day 49 and 56. And you can see that the efficacy of the vaccine continues to increase. So your CD4 cells are picking up this vaccine, making good neutralizing antibodies, and continuing to make a good immune response for this vaccine. Next slide. So more good news. What does it prevent? Death. Preventing death is a very good thing for a vaccine. And this vaccine, much like the Pfizer vaccine, much like the Moderna vaccine, is excellent in preventing death and in severe cases. So it really prevents hospitalization and death. So it will decompress the hospitals significantly as we get this vaccine out there. Asymptomatic, there was some hint that it decreases the amount of asymptomatic disease, but in reality, it's difficult to say because there weren't enough numbers in what was presented to the FDA in their presentation. Next slide. So what about people over 60 years of age? And if you have significant, without comorbidities, the vaccine is very good. So if you're not diabetic, if you're not obese, if you don't have COPD, if you don't have heart failure, vaccine works great. So if you're a healthy 60-year-old, it 100% of the time prevents severe and critical illness and moderate to severe illness. Moderate severe illness meaning tachycardia, increased respiratory rate in their definition. With comorbidities, it is not as effective. And in severe critical cases, again, it is not as effective as in preventing disease. It doesn't mean you're going to die. It means that you're going to become a little bit more ill. Next slide. Next slide. So the safety. So the safety analysis through January 22nd, and it was randomized, showed it was a very, very safe vaccine. You did have a numerical imbalance, minimal numerical imbalances for people who had thromboembolic events, tinnitus, angioedema, wheezing, arthritis, and peripheral neuropathy. But the FDA felt this was really a vaccine reactogenicity and not a major side effect of the vaccine. Next slide. Dr. Lynn's important thing, which is pregnancy. Like I said earlier, this vaccine platform, because it was used in Africa for Ebola, so the platform itself has been shown to be safe. 8 pregnancies were reported through January 22nd, 4 vaccine and 4 in the placebo. In the 7 participants, vaccination was within 30 days after the last menstrual period, and 1 vaccine recipient was prior to last menstrual period. And unsolicited adverse events related to pregnancy, including spontaneous abortion, 1 in the vaccine, 0 in the placebo, incomplete abortion, 1 in the placebo, elective abortion, 2 in the placebo, and ectopic pregnancies, 1 in the placebo. Among the participants in the vaccine group, 2 pregnancies were ongoing with outcomes unknown at the time of presentation. So nothing can be said about pregnancies. It combined, and then in rabbits, it's been shown that it is safe, and there's no data on pediatric population. Next slide. So there was a question about country-specific rates and variants. And so the country-specific rates are as follows here. So in the United States, moderate to severe critical illness, the vaccine efficacy is 74% at 14 days and about 72% at 28 days. In severe critical illness, again, 78% efficacy and 85% by 28 days. So you get to South Africa, okay, it falls off significantly for vaccine efficacy, 52% because of circulating strain. But you have to remember, okay, it didn't prevent the illness, but it prevented death. And if death is a pretty good hard endpoint, because the way they define moderate to severe critical illness is a little different than what we as doctors think of moderate to severe critical illness. So it's not so great at preventing moderate illness. It's better at preventing severe illness, and the vaccine efficacy is about 81% against what they define as severe critical illness, and it's 100% efficacious against preventing death. In Brazil, with the P1 strain out there, it's about 66% efficacious in preventing moderate illness, and it's 81% efficacious at 14 days. And then when you go out 28 days, it's 68% efficacious at preventing moderate and severe illness, and then 87% efficacious at preventing severe illness. So the vaccine works with 1 single dose, doesn't need significant refrigeration, easy to transport, and so you do now have a vaccine that is a game changer because it is safe, it's easy to give to all populations, and you don't need all of these gymnastics of bringing somebody back a second time and timing it and storage. Next slide. So the summary of the J&J vaccine: it decreases COVID infections in all populations tested, including race, ethnicities, comorbidities. It probably decreases severe infections in this number. It's safe, well tolerated in all populations, and young people probably they have better antibodies than older people, and so they have a little bit increased side effects as compared to the older population. And there's no evidence of antibody enhanced disease like you get in dengue vaccine. So really nice, good, safe vaccine. Next slide. So what are the unknown concerns? Efficacy against asymptomatic infections. This does not produce sterilizing antibodies. There's a trend that says it may decrease the amount of viral carriage in those. That's to be determined. It has decreased efficacy in patients over 60. Minimal data in HIV patients. I had about 600 people. No data in children. No data on long-term side effects because it's brand new. Minimal data on duration of immunity. And the durability of a single dose is unknown. There are ongoing trials with phase 3 trials going on that are looking at a 2-dose vaccine, and that'll be out soon. Next slide. So another piece of really good news about vaccines here. This is published in the New England Journal yesterday, and this is basically a Pfizer trial. Israel has basically a single system of healthcare. There are 4 big HMOs that take care of most of the population out there, and they had a real-world trial that looked. It was an observational trial, and it was a very short study, 14 days, and they looked at the vaccine being given to their general population and how efficacious it was. And so, next slide. Basically, it showed, and this is the randomization. You had about 600,000 people. You don't have to read this whole thing. I would advise you read the article or listen to the audio portion of it. So it had 600,000 people who were eventually randomized to this. And in the real world, it showed that it was really no different than the trial that was done as a placebo-controlled trial. The efficacy in all age groups was well over 90%. The side effects were not significant. There was, no huge amounts of Bell's palsy or anything else that went on. Next slide. And there were very well, the matched groups were good, and it showed that this vaccine really works just like it worked in the trial. So we expect the same thing to happen with Moderna. So these vaccines really, really do work. And I think if we get enough vaccines into arms, as you'll see the rollout in the next few days, we should be able to get out of this. The rapid drop in cases that we're seeing now is a consequence of all of the death that we had over the last 2 months. As we relax these, the measures that are going on with gatherings, you're going to see, again, a small bump up in rate. As the weather gets warmer, I think we're going to have a significant drop in rates, and it's going to be a better summer. And hopefully we'll be out of this by July, August, so we can have some sort of normalcy. Next slide. And this is the cumulative incidence of 5 outcomes, the severe COVID disease. So it shows that it decreases the documented COVID infections. So Pfizer does work to decrease actual transmission. It decreases the symptomatic cases significantly. It decreases COVID-19 hospitalizations. It significantly decreases the severe COVID-19 cases, and the death rate plummets when people are vaccinated. So the vaccine works as advertised. Next slide. And so we'd hope you continue to use the Trusted Messenger and go to the vaccinetrust.org. Next slide. And Vaccinate Chicago, where we do have a significant amount of volunteers out there. We've been reaching out to the health departments to best deploy these, and there's going to be a significant change in the next few days of how we vaccinate folks. So hopefully we'll be able to participate in this and get this behind us. So I'd like to thank the Chicago Medical Society for putting this together. We have Dr. Tariq Butt for a few comments. There are a few chat questions out there about the current graphs about herd immunity, assuming vaccines will be effective against all new strains. And that's actually true. The assumptions are assumptions, and the assumptions change. If we have if we have new variants that don't work against the strains, then we're going to be in trouble. But the J&J data is very good in the sense it shows that the deaths were prevented. Hospitalizations decreased with the J&J vaccine, and it was done in South Africa. It was not as efficacious to prevent mild disease, so you may have gotten mild disease, but you didn't die from it. So that's a good thing. Next, the question was, the current vaccines appear to provide effective humoral immunological effects for at least a year to date. The data are out there. I believe it does provide good immunity for long periods of time, and including neutralizing antibodies. The antibodies that we check now, which are binding antibodies, don't correlate as well as neutralizing antibodies, but they do seem to give you a significant marker that you have uptake the vaccine and you'll be protected. And then, what is a recommendation for vaccinating patients with inflammatory disease, and is there a minimum time between vaccinating after stopping? That's a great question. We actually went over this as a group in our group. So there is a panel out there of rheumatologists, including 6 infectious disease doctors, like a 30 people panel. And basically, they say that if you're on methotrexate, you should hold the methotrexate for a week prior to giving the vaccine. It's actually quite complicated. If you could just email me, I will get you the recommendations because it's quite a long table. We have that put together already. And safety profile for pediatric populations. Are there any reported issues for that population, Dr. Lin? Because I know they're recruiting and there's trials in pediatric populations. I don't know the data for that. Would you happen to know? No, zero data. Okay. And where can we get vaccinated? That's a great question. I think the Biden plan is going to change where we're going to get vaccinated. We're going to have mass vaccination centers. The states are also going to be participating in this. So we'll have more news on that. And Dr. Butt, who's the president of the Chicago Medical Society, if you can say a few words, Dr. Butt, for getting all this together. Thank you.
Thank you, Dr. Chandy and Dr. Len. You are an amazing panel, and good evening to all of you who joined us. I'd like to thank you on behalf of Chicago Medical Society, and I want to say that if you're a member and you renewed your membership, thank you so much. If you're a member who needs to renew membership, please do so. And if you are not a member, please become a member and join our Trusted Messenger campaign. And also Vaccinate Chicago, as Dr. Chandy mentioned and Dr. Len mentioned, we need to vaccinate a lot of folks, and we'll try to help you become a volunteer, become part of a vaccination team. And also, as Dr. Chandy mentioned, we'll make sure that you also have information about where you can get vaccinated. Thank you so much for joining us. Thank you, Dr. Chandy and Dr. Len, again, for a fabulous presentation.
Well, thank you. There's one other question here that we get questioned a lot of times. Any concerns about thrombocytopenia risk with Pfizer or Moderna? There are reports of ITP with these vaccines, but they do not seem to be above the background. And then there's a question about NSAIDs with Tylenol around vaccination. And what about seniors on low-dose aspirin? The low-dose aspirin should be continued. And then taking acetaminophen around vaccination should really not change the vaccine efficacy at all for this.
Right. The recommendation really came from the pediatric world because years ago there were studies that showed that when Tylenol or ibuprofen were given prophylactically, it may have slowed the immunologic response to the vaccine. So it's not recommended to pre-medicate, but if patients become symptomatic after vaccination, acetaminophen appears to be appropriate. If you have little pyrexia, or if there's difficulty with myalgias and headache, that should be appropriate. But the pre-medication is what we're suggesting to avoid.
Yeah, you shouldn't pre-medicate for most of these things because you're giving a drug without symptoms, which doesn't make sense. Well, thank you very much for joining us. And I'd like to thank Chicago Medical Society and for all of you for participating. And hopefully everyone's vaccinated and we can get through this in a few months. Thank you.
Thank you.
Stay safe out there.
Yes, stay safe, please.